things that I'm doing in the lab that I'm currently having a lot of fun with. So whoops. So straight from the lab website here, what does my lab do? Right now, we have several major funded projects that involve things like developing reverse vaccines. I'll explain what those are that target invasive arthropods, in this case, Amorphosis longicornis. Genotyping spatiotemporal distribution of arthropod-borne disease agents, whether they're viruses or bacteria or otherwise. Vector-borne pathogen surveillance and discovery, so this ties in with bullet point number 2 there pretty closely, as well as molecular evolution, in this case of arthropods. And that'll be the final project that I talk about. So I'm going to start off with the USDA NIFA funded work here to develop a reverse vaccine that targets the Asian longhorn tick, Haemaphysalis longicornis. So the premise of this entire work is to deliver an antibody to the tick that decreases its own fitness. And since the tick itself is basically just a big syringe, it'd be great if we could utilize the host to deliver that antibody, which means that we need to prime the host immune system, in this case cattle, against the tick. So what we would do is immunize the host with some molecule that was derived from the tick, whether it's from salivary glands or midgut or some other presented molecule that is antigenic in the cow, and the cow would create antibodies towards this protein, and then the tick then would feed on the cow, take those antibodies into itself, and then have some phenotype because of that. So we screen tick genomes for these proteins, and to do that, we first have to put tick genomes together, and that's the crux of the matter that I'll talk about in a second. So those immunoglobulins, those Igs, are taken up by the tick during the blood feeding process. So in contrast to acaricides, which are the most commonly used control measure for ticks right now, anti-tick vaccines are the least. So they're on the opposite end of the spectrum. And acaricide always comes with the specter of acaricide resistance, so it goes without saying that we need some new and novel control means for these arthropods and basically any other arthropod, right? So this discipline isn't unique to ticks. Other blood-feeding arthropods are subject to research also. There has been some work done on the market. Some of it has gone to market, specifically with things like the BM86 midgut antigen. This was marketed as tick guard against the cattle fever tick, Boophilus annulatus, Rhipicephalus annulatus, you might recognize it as. So its efficacy was promising at first, but there wound up being too much variation in the epitope within the tick throughout different tick populations for the vaccine to be effective throughout the country, specifically most of the southern United States. So it worked in some places where there were particular haplotypes of this epitope in the tick, and it didn't work in other places. So it's kind of fallen out of favor in the market. However, the advent of next generation sequencing, specifically high throughput genomics, has given us this opportunity to screen genomes pretty quickly for the proteins that they encode and then select these proteins based on certain characteristics as to whether they may or may not be antigenic in a cow, for example, and create antibodies that could be detrimental to the tick. And this has happened with several strains. So there are ticks, and there are also there are ongoing projects for the stable fly right now that involve this, as well as some preliminary work in Haemaphysalis longicornis as well. They were taking proteins that they already knew existed in the tick based on proteomics and not genomics. So as our application here to the ALT, which probably doesn't need a lot of introduction in our sphere, it was discovered in New Jersey in 2018. Gone to the latest stakeholder meetings, unfortunately, over the last few months. Hundreds and hundreds of counties throughout the United States, causes extreme infestations, and most importantly, it vectors a particularly pathogenic strain of Theileria orientalis, Ikeda genotype. But to do all this, the first thing we need is a genome. And that's what we set out to do. So that was the ground floor of this entire project. So to do this, we use PacBio HiFi single molecule sequencing coupled with Illumina Hi-C chromosome conformation capture sequencing. So HiFi will give you very, very large single molecule length reads or pieces of DNA rather than fragmenting the genome into lots of tiny pieces and trying to put it back together, which is difficult. We get, 6, 7, 8, 10, 12, 20,000 base pair reads out of this technology. High-C chromosome conformation capture basically just cross-links DNA with itself and then rips out pieces that are close to each other on the chromosome so you can use those to scaffold your genome later on. I could give an entire talk about how these two technologies work, but for now, this is all that I'll say to save time. So the assembly here was really challenging because the tick was triploid. Every assembler that I've worked with has been built and has algorithms that deal with diploid assemblies, which means it's unzipping two haplotypes for you and giving you essentially a haploid genotype and then an alternate haplotype. Having a triploid tick really confounds the assembler in ways that I won't get into, but it took quite a while of purging, unzipping, removing what I knew was one haplotype, keeping the other two, reassembling the data, removing a haplotype. We ran a couple of utilities several times over and then reassembled the tick genome over and over again until we were quite certain that we had only one, or rather we had a diploid assembly of which we can take one haploid or one strand of DNA and put it forth as being quote unquote the tick genome that we publish and that we use for working with. This single sentence on the slide right here encompassed probably 3 months of work trying to get at a reasonable genome assembly for a triploid organism, which is pretty difficult, because there are 3 alleles at any single locus. But what we got was very good. Over the course of our project, there were a lot of Longhorn ALT genomes deposited into NCBI as this tick kind of went global and everybody became interested in it. So what I'm drawing your attention here to in the yellow is the reference genome assembly from Gia et al., or which was at the time the reference genome assembly. It is assembled to chromosome. Our assembly was not assembled to chromosomes. It was in 270 scaffolds, which means we had the 3.1 gigabase. So it's basically, almost the size of a human genome. 3.1 gigabase tick genome and 270 scaffolds. G et al. Did have it in assembled to chromosomes, but they also had a lot of extraneous scaffolds as well. The problem with that assembly, if you look here, was that it encoded, or they were only able to identify, I think you guys can see my cursor on the screen, right, that I'm moving here. 53.6% of what should be a complete arthropod proteome was identified in their assembly, and that number is very low. Now that could be a lot of things. Maybe their RNA-seq wasn't very good, their gene model prediction pipeline wasn't very good, or their assembly was just full of ends or had been scaffolded very heavily and most of the genes were actually broken. If you look down here at our assembly, within the genome, the assembled genome, we had 94.4% of an arthropod proteome and 95% in our protein prediction. So despite the fact we didn't have a chromosome level assembly, we had a really good assembly for terms of gene finding and actually doing some analyses that we needed to do with this tick. So we did a lot of RNA-seq, tissue-specific RNA-seq from ovary, midgut, salivary glands. That's what you see on the left there, coupled with a long list of informatics software that scored the proteins that we had predicted on the tick genome based on their likelihood to be antigenic in a cow. Some of those included things like vaccine. Vaccide. I trained Vaccide with parasitic immunogens from other work to try to determine whether or not something might be immunogenic. We also predicted the major histocompatibility complex 1 and 2 epitopes that were on each protein, as long as B cell epitope scores. And at the end of the day, I had a very large spreadsheet with some summary data on it that I used to decide what to move forward with, because we really only had room to test one antigen here in a cow. So this tick had, a proteome of something like 20,000 genes, and I had to pick one. I had to pick the best one. So I looked at 4 to begin with. These 4 had high expression across several different tissues. They were highly scoring as far as machine learning algorithms went. That was annexin. So annexin forms gap junction channels and hemichannels between cells, facilitates transmission of endocrine signals between cells. Follistatin was another one. This is involved in cell proliferation and differentiation. It has been used before in some RNA knockdown that caused a decrease in tick oviposition but didn't affect the blood feeding process. Spark functions in oogenesis. It regulates follicular cell mitosis, maintains cytoskeleton integrity. And VDAC, which was suggested to me by a collaborator, Glenn Scholes at the USDA, was being worked on in some other ticks. So this is an ion channel in the mitochondrial membrane, the outer membrane, which transfers ions and metabolites in and out of the mitochondria. And so there are trials in Rhipicephalus micropus that are underway right now. So of those four, sorry, so I should introduce the fact that we checkpointed our study at this point, and we published the genome and the informatics screen for our antigenic candidates. This was led by Mohammed Mustafa, my postdoc at the time. So Mohammed is now an assistant professor at the Kansas State School of Veterinary Sciences, where he's doing all sorts of other fun stuff, including continuing this work and others. We moved to an RNAi stage after this. So rather than just screening candidates in stall trials and testing whether or not they may be antigenic through something like ELISA, we decided to use RNAi first. So we generated synthetic constructs for those 5 genes of interest, created double-stranded RNA from them, microinjected 1 × 10⁻11 copies into the hind coxa with a microinjector that I bought as part of the project. Control ticks were injected with the injection buffer only. We injected 4 groups of 40 adult female ticks. 30 per group we put on rabbits. So this was to test the overall efficacy of the RNAi knockdown and the phenotype that it might impart on the tick. And then we saved 10 for follow-up RNA-seq to QC gene knockdown. So this leads us to the rabbit stage of the project, which is my least favorite stage of anything that I've ever had to do here because it involved animals. So our affectionately named Chrome and Firefox, so the vivarium folks named them for us, were part of our infestation trial. So we had our control ticks, our annexin injection ticks, follistatin, and Spark. We did VDAC separately because at the time I could only afford 2 rabbits without mortgaging my house. So there were 30 ticks per group on each ear. So we used ear bags. Special thanks to Alvaro for explaining and showing us his protocols for feeding ticks on ears. So these were the micro-injected ticks. Of which not all survived, and this is common in tick experiments. It's kind of disheartening to go in and see a bunch of dead ticks in the ear bags the next day, but they don't all find a means to attach, and they'll desiccate pretty quickly without a very high humid environment unless they do attach quickly. Some of them even got stuck in the ear bags. So I think if we were to do this again, I might actually deposit the ticks in a different manner. So we had 19, 21, 23, and 22 survivors across all 4. And in this case, the tick was the replicate, so we were still pretty confident in this. So what did we see out of this? At the end of the day, we left the ticks on there until they were fully engorged and dropped off, and then we went and collected them, and we put them in individual test tubes, and then monitored their oviposition and several other life parameters beyond that. So the first thing that we noticed here was that the feeding time in days of the Spark injected ticks was much higher, right? So the feeding time was just time to detachment. So most of them had detached after 6 or 7 days. Nearly all of the Spark ticks stayed attached for an extra day and detached at the end of day 8. So we checked them twice a day. This was in the mid-morning and then in the late afternoon. The engorged weight, not much difference between the control and the follistatin. The annexin and the Spark injected ticks were smaller or lighter or less engorged at the end of the day than the others. So you can see significant differences up at the top there. But really, here come the big differences in the annexin injected ticks. So the pre-oviposition days were the number of days that they held their eggs before ovipositing. So the 3 other conditions kind of all oviposited on day 6 and they were done with it. The annexin injection ticks held their eggs in some cases until day 19. So we were all getting sick of going to the vivarium twice a day for like 3 weeks to check, or not the vivarium, but over to the lab to check on how many eggs had been produced. And these annexin ticks, for some reason, just held their eggs for an extremely long time. As well as the fact that they produced less egg mass than the rest of the trials here. So there wasn't much difference between control and follistatin significantly. Spark did produce fewer eggs. The annexin injection ticks produced far fewer eggs than the other conditions. So based on this work, and I will loop VDAC in, if you remember VDAC was that 5th gene I talked about, We got no knockdown of VDAC, and I don't know why that is, so I just excluded it from the entire analysis. And now that I say that, I'm realizing I forgot to put in the FPQM quantitation for the knockdown efficiency. We have full body RNA-seq for these ticks here, so we can actually go through and look at exactly what happened metabolically to the tick based on that RNAi knockdown, rather than just quantifying single gene knockdown. So we also, with the annexin injected ticks, noticed that their chitin or their wax coating kind of started to slough off. They all got some form of dandruff. Unlike any of the other ticks, and like I said, this is a gap junction protein, so it is responsible for cell-cell adhesion. So it's quite possible that had some kind of effect in the epidermis that made the tick more susceptible to sloughing its dermis. I don't know exactly what or why that happened yet. We have these frozen. I may go back and look at them later, but it was an interesting side note. So where do we go from here? Annexin is going to be the candidate moving forward. So right now we're synthesizing recombinant annexin protein at Kemp Proteomics for us. So they're going to do the synthesis for us. The vaccinations are going to be conducted at USDA Pullman, Washington in their animal stalls because we don't have the capacity to do that here. We designed the experiment with input from some superheroes that kind of rose like a phoenix to save the project after we lost some other folks that were able to do this. Karen Poe, Reggie Bastos, and Susan Ney out at USDA Pullman all agreed to take care of this for us very, very generously and contribute their time. So we're going to have 4 trials of annexin vaccinated cattle and then 1 control. So we do only have a 4+1 in this case because the animals are horribly expensive. They're limited for resources. And nowadays, there's enough of this stuff being trialed that unless we see a severe effect, it's probably not worth moving forward with. And I think we can do that with a 4+1. So those injections are going to happen summer 2026. We'll track tick fitness as prior, and we'll also assess antigenicity in the cattle to make sure that they actually developed antibodies to this gene via ELISA. Okay. Does anybody know how to erase that little red thing that I think somebody drew on the screen? And I don't know. It'll be there for the rest of the talk. I apologize. But
I'm trying, Dana.
if anybody wants to erase it, feel free.
I don't know how to do it. I noticed.
Okay. Um, so where do we go from here with this project? So one is that the project is now being wrapped up out at USDA Pullman. Um, the future is clearly, I think, and based on COVID, going to be mRNA vaccine technology. So this allows rapid screening of a lot of multivalence rather than having to generate lots of recombinant protein and vaccinate cattle with it. We want something that's compatible across multiple tick species. And having artificial feeding, and I think Chang Li said Alvaro was teaching, but I'd be pointing at him, and I don't know if Jenna is on here. Having an artificial feeding system in place so we don't have to go use the gosh dang rabbits would be amazing. So we can trial some of this via artificial feeding system and at least see whether or not our recombinants have some effect on the tick before we actually test to see whether they're antigenic in cattle and then whether they create an effect after blood feeding. So artificial screening, I think, or artificial feeding is going to be fantastic. They haven't developed it yet for longicornis. A lot of people are trying. It's kind of so-so. It's not where we need it to be yet. But I have this micro injector and I spent all the money on it, so there's no reason not to do this in other arthropod species as well. I've floated this for things like midges and mosquitoes and so on and so forth. So we can have a lot of fun with this moving forward. Okay, so before I leave ticks entirely, I'm not going to recapitulate Zoe's PhD because she did a great job of that herself. And actually, Steph, I'm not going to talk about yours either. Sorry. I'm going to let you guys, the grad students, introduce those, but I will throw out all the great work that she did on her way there. And her 4th paper, which has been under review in Journal of Medical Microbiology for 4 months now, and they're still working on trying to find a 3rd or 4th reviewer. So ASM really needs to get its act together. It's driving me crazy, but I'm pretty confident in it. What I will do is talk about all of these wonderful samples that Zoe left in the freezer. Tens of thousands of ticks that she collected throughout New Jersey as part of her PhD work and then surveilled for various pathogens are now mine to do whatever I want with. So the question I'm asking here is what's in our ticks, or rather, what is in our ticks that we don't already know about that Zoe has tested for? And what you're seeing at the bottom of the screen there is a CHIME taxa plot of all of the wonderful bacteria that are in 633 Amblyoma americanum species. It goes off the end of the screen there. Um, so, like I said, tens of thousands of ticks. We started with some amblyoma. We started with enough that I could actually have money to sequence, and then we went ahead and started looking at the bacterial community compositions that were in these ticks. One of the things that jumped out right away, at least for me, was Candidatus midichloriae mitochondriae. So this is still Candidatus because it hasn't been cultured in the lab. It hasn't successfully yet. This is an obligate endosymbiont in lots of ticks, specifically Ixodes ricinus. It's a mitochondrial energy parasite, and it invades between the outer and inner membrane of the mitochondria and devours the mitochondria. Can be horizontally and vertically transferred between ticks and among ticks. What I saw in our data, surprisingly, were two completely different OTUs or operational taxonomic units of Candidatus midichloriae mitochondriae. I'll call it the gray OTU and the green OTU. So there were 46 of the 633 had this bacterium. And this was on my radar not only because it was an endosymbiont that I had known about, but because it's also now a potential pathogen. So this was isolated and sequenced from an eschar from a pregnant female in Tennessee that resulted in loss of her first trimester pregnancy. So whether it's an endosymbiont or a pathogen has always been a major question for me. And when I saw two major OTUs here, and these are separated by 11 fixed nucleotide differences, I compared all of them with each other. They are quite different. What I'll call the green OTU here seemed to be the canonical endosymbiont. Its top hits were all to Candidatus midichloriae endosymbiont of XYZ. So this was our amblyoma endosymbiont. But moreover, what I'll call the gray OTU had top hits to Rhipicephalus sanguineus from Thailand, Amblyoma lepidum from Africa, Hemiphysalis wellingtoni from Thailand. None of these were from the United States. So this was a little bit confusing to me until I went back and actually read that EID paper that described the pathogenic bacterium isolated from this female. And they say here in the resulting tree, we detect the bacterial sequence had the highest homology with a sequence from Thailand, KY910125. Well, look at the top of the list. KY910125. This is the same bacterium putatively that they isolated from this patient out in Tennessee. So it appears that here in New Jersey, we have an endosymbiotic Candidatus midichloriae and a potentially pathogenic Candidatus midichloriae commingling. I found none of them in the same tick, so they appear to be what's the word I was looking for? Separate from each other. They're not overlapping. I'd like to follow up on this to a much greater degree and try to get some genome sequences out of these bacteria to identify, maybe the mechanisms of pathogenicity versus endosymbiosis and so on and so forth. But I think this is something worth surveilling and following up on, giving its potential status as a pathogen. We also detected Mycoplasma cynos. So this is a canine respiratory pathogen. Mycoplasmas can be transferred by ticks, but I have not yet seen cynos on the list of tick-borne pathogens. Cynos is usually dog to dog. It leads to things like kennel cough, but it can be pretty severe. The role of ticks in transmitting respiratory pathogens is unknown. It's unlikely that they're going to deliver it to the respiratory mucosa where it can actually take foot, but after we published this paper, I got a call from a medical doctor out in Wisconsin, and he said, I just saw your paper. I want to let you know that we had a patient out here that was covered in ticks, had lots of tick bites, went camping, and then had a lot of joint pain. We thought it was Lyme borreliosis, and one of the things that they actually do is aspirate synovial fluid and then sequence it to see what's in it, which I think is really cool. And he said it was actually Mycoplasma cynos that we detected in this patient's synovial fluid. Do you think that could have been tick-borne? And I said, that's really awesome. If you want me to look at the actual sequence to see, I'm not sure if medical doctors are up and up on the informatics and how to determine, top hits versus some secondary or composite homology. But I'm waiting to hear back from him to see, but, we might be onto something here. So this is all stuff that's starting to fall out of our sequencing of ticks. I haven't touched, what may be the most important tick in the bunch, and those are the 4,000 plus scapularis that Zoe collected in her time here and tested for standard tick-borne pathogens. But I think we can have a lot of fun doing this with our different tick species throughout New Jersey and learn a whole lot about what we don't know about our ticks. So this remains to be analyzed, given funding. Okay, so moving on from ticks to mosquitoes here. Um, I introduced this project again during my candidate seminar. This is the use of mosquito excreta as a template for genomic epidemiology, right? So what do we do when you want to know whether polio or COVID or something like that is endemic in a population? Is you go to the wastewater. So in our case, we built mosquito toilets. And we use them to do lots of fun stuff. And this is predicated on the fact that as mosquitoes feed, they'll salivate as well as excrete. And if you can get them to salivate on something like a filter paper, you can keep that filter paper and test the saliva. And if you can capture the excreta, you can test the excreta for various things. So this isn't a new technology. People have been using this for years now to isolate pathogens and to test for things. Um, but what we haven't seen yet was a really efficient way at collecting the mosquito excreta. So that's what we set out to do. Um, because really, it goes without saying, and this goes with what I just talked about in regards to ticks, you don't know what's here unless you look for it, right? With a rapidly changing vector-borne disease landscape, collecting tens of thousands of mosquitoes and grinding them all up, and then searching for an 11 or 12 kb viral genome in a stack of 100 mosquitoes that have 1 to 3 gigabase genomes can be pretty difficult. Aside from using qPCR, we would like to use metagenomics to do this so we can move into the realm of pathogen discovery as well. So our first excreta collecting device was this funnel. This was printed at PLA. Taewon Han printed this over in environmental sciences. In conjunction, our collaborators were Mehdi Jevanmard over at the School of Engineering and Jolun Meng, his graduate student. And you can see the ice cream container there that's sitting on the right has 50 Culex mosquitoes in it. There's a dental dam on the top that's soaked with sugar water. It's colored red because it has some food dye in it. And the mosquitoes are excreting and it's running down the funnel. So the funnel is coated with a proprietary super hydrophobic spray that cost a lot of money and a lot of time and money to get here from Japan. Taewon swears by it. He says it's the best spray that he's ever found on the market. So we sanded it with a fine grit sandpaper, baked the funnel, coated it with a spray. And you can see at the bottom here, this is actual mosquito excreta from the 50 Culex mosquitoes that are in that ice cream container up at the top. So we can take that then and then prep it for next-gen sequencing. So the proof of concept work was actually done quite a while ago, almost embarrassingly a long time ago, 2021 and 2022. I'm just now talking about it because I've been sidetracked with so much other stuff, but I can say it's done and it's been submitted. It's under review now. This was nothing but proof of concept of what we can do with this data, and we did some really fun stuff with it, right? We're trying to develop means to assess the RNA virome of mosquitoes. So we had these two pools of 50 Culex mosquitoes. We collected them right behind Headley. We just walked out in the backyard and put a gravid trap out there. So they were gravid female mosquitoes. Gave them sucrose, let them excrete for 18 hours, and then sequenced total RNA. And there was a lot more involved than just this, but for the sake of time, I'll condense it down. There was involved a whole transcriptome amplification step and so on and so forth till we could enrich our RNA. And what I decided to focus on here were these 19, and I'm going to call them full-length viruses, or at least viral segments, if it's a segmented virus, based on the length of the assembled scaffold in the data. So this underwent, short read assembly. This was Illumina data, short read assembly, just like I would with anything else. And then homology searches against NCBI nucleotide and protein databases. So I had to decide on what to talk about. I couldn't write a paper about 400 different viral fragments and what might be a virus. So I chose what was complete or nearly complete here. And I divided them down into what are ultimately 19 different viruses that you can see here. The percent similarity column in most cases is pretty high for those, which was made it a lot easier to focus and describe some of these, and others it's low, and those I would describe as putatively novel viruses. Whether or not they were novel, I have yes, yes, yes here. Those I described as novel in NCBI. Whether they were present in funnel number one, number two, or both. I should say these samples were taken 1 year apart, which is about as fast as I move nowadays. But what I want to talk about are a few things here. Those that are highlighted in yellow were what I described as putatively novel. A Partidi-like virus and picoRNA-like viruses. So these are both rapidly expanding lineages of viruses that we're finding in more and more arthropods. Some of the Partidi-like viruses do include pathogens of things like arthropods and I believe nematodes. I can't remember. I'm not quite sure. They have a very wide host range. I also detected a novel Leash-Boo virus, which a year ago was called Leash-Bunya virus. It's a lineage within the Bunyaviridae that infects protists and flagellates. Not mosquitoes, but it made perfect sense because our samples were full of different trypanosomes. And I was worried about time, so I didn't put a phylogenetic tree in here, but I did take all of the trypanosome 18S small subunit ribosomal RNA sequences and put them in a trypanosome tree of life. And we had lots and lots of different representatives throughout different trypanosomatid genera in these samples. So these mosquitoes were full of trypanosomes, and the trypanosomes themselves were at least some of which were affected with Leash-Boo virus or Leash-Bunya virus. So this is two-segmented. It has, or I detected at least two segments in there, the large and the small segment for those. Um, so really some of the coolest stuff that rolled out of this was one Hedwig virus. So I'm calling this new to the Americas because it is. So this is the first detection of Hedwig virus in the Americas. It's only been described from Europe thus far. Homology was very high. It was 96, 97% amino acid across the entire large, medium, and small segments. So it's a tripartite virus. Hedwig existed initially as only a small voucher sequence from a dead snowy owl in Europe that was isolated concomitant with West Nile virus mortality. So it does putatively have the potential to infect vertebrates. It was placed recently in the novel genus Griffinivirus. So if there are Harry Potter fans, you probably recognize Hedwig and Gryffindor and so on and so forth. So whoever, the virologist was that worked on this was a Harry Potter fan. We assembled complete small, medium, and large segments from the tripartite viral genome. The first detection outside of Europe. And I've also found this in several other samples in New Jersey that I've been toying with. They're not ready for complete presentation or deposition yet. All of these have been deposited to NCBI now and are in the associated manuscript. Also, new to the Americas, Ahes virus. Not much is known about this. This is a totavirus. Totavirus is a very large group of viruses. Thus far, AS virus has only been isolated and described from Culex metagenome samples in Sweden. And what I thought was funny about this was that the authors that worked on this postulated that it was actually an environmental contaminant. They say here Ahes virus and totavirity was at low abundance, not present in several libraries, and clustered with viruses derived from various environmental samples, suggesting that it's not mosquito associated. So we have pretty good data here that suggests it probably is mosquito associated. We detected it at 150x coverage. That means there was a lot of deep sequencing associated with this virus in the excreta. Now, it obviously could have picked up some kind of external contamination if it touched the scales of the abdomen or something as the excreta was being deposited. I don't know. But I think we have pretty good evidence that it's a mosquito-associated virus at this point. What I will call second detections in this case, Black Hawk Lake virus. It's been detected once in the United States in Iowa from a Culex metagenome. This is a completely unclassified virus. We know very little, if anything, about it. I found this several times since then in our New Jersey Culex mosquitoes as well. So where do we go with all of this? So it took me, I'm going to say embarrassingly, it took me, I don't know how many years to write up samples from 2021 and 2022 that only included two samples and two funnels. We now have 63 samples throughout New Jersey that have been collected of mosquito excreta using this funnel to analyze. Don't ask me how I plan on doing that. I'll try to figure it out and present the results in a way that doesn't involve a 400-page research manuscript, but this is really a lot of fun for me. It's determining stuff like this. Again, I think if you don't look for it, you're just not going to find it. And I think there's a lot of diversity out there that we just don't know exists. And we're ready to hop on that, at least with regard to New Jersey. So these samples targeted different habitats, different mosquito species from 9 different counties throughout New Jersey. So we have a very broad sampling plan here. Now, last time I kind of brought a mock-up of what actually exists as a mosquito excreta collecting trap now. I think it was like a pickle jar that had my outlet at the bottom and an inlet at the top. This is actually, I should have taken a photo of the entire 3D printed object, but both of these traps actually exist. One is a downdraft trap that would suck mosquitoes in down through the top into an inner chamber. The air would move along the outside of the trap and then down out the outlet, while the excreta would be sequestered in the inner funnel here. So it turned out the answer to our problem, which was how do you collect these tiny drops of excreta in a trap that has this fan torrential air current moving, is to just put a funnel within a funnel. So we have this funnel inside the trap, as well as an updraft trap, which is kind of like a big bell, and it sucks the mosquitoes in through this ring around the middle here, and they go up into the inner chamber and then excrete into this funnel here. These two holes on the outside are where we plug in a pair of urinalysis cups that have a wet sugar-soaked sponge in them. The mosquitoes feed on the sponge and then excrete into the funnel. So we trialed this a few times, and it does work reasonably. We don't get nearly as much excreta as we get when they're sitting in a lab overnight with no air current. You have 50 mosquitoes in this tiny little ice cream container with nothing but a funnel underneath. But we do collect excreta droplets in the attached Eppendorf tube the next day. I think our biggest problem will be evaporation. We'll be fighting evaporation because when those sugar-soaked solution droplets evaporate, they kind of get sticky and they impede the flow. And mosquito scales can also stop things from moving down the funnel when they get shed. I do briefly want to mention bacteria in here as well as viruses because we also wrapped this up and published it as well for folks that remember Miranda. Miranda is now a presidential fellow PhD student at University of Florida, Gainesville. So we just took some of our excreta and we plated it and we grew bacterial colonies to see what was going to grow there. And we had some fun sequencing complete genomes of mosquito-associated bacteria from that. We were able to publish one in particular, which we knew was Serratia marcescens because it turned dark red, even though the editors at MRA didn't let us say we knew it was Serratia because it turned red. We could only say it looked like it produced prodigiosin. But this was the second smallest fully assembled Serratia marcescens genome reported, and whether or not that may come with the mosquito-associated life cycle, it doesn't appear to have a smaller gene content. It still had lots of genes. But it was the second smallest Serratia genome reported, and we got that wrapped up and published as well. So we do have others that are frozen at -80 that we can resurrect and then do the same thing with in the future. So what I want to finish off with here was a project that for me is just a lot of fun. Um, I could have talked about pretty much anything to close this out, but I see myself doing more of this in the future, and also because it involves our Rutgers Entomology Museum. So this was also a George H. Cook project that for folks that remember Taito Jenkins or Tahila Jenkins, our undergraduate who moved on last year, this was Sir George H. Cook. She entitled it A Blast from the Past, Comparative Analysis of Extant and Historical Genome Data in Anopheles crucians and Anopheles bradleyi. I'll just call it the Anopheles bradleyi Genome Project, or rather the Anopheles crucians Genome Project. This was funded by the New Jersey Mosquito Control Association Research Fund, which we are eternally grateful for. So Anopheles bradleyi year over year tends to be flip-flops between the second or the fourth most abundant mosquito collected in New Jersey light traps. This is a salt marsh anopheline mosquito, comes off by the thousands and thousands and fills traps year over year for those at the coastal communities. It's part of the Anopheles crucians complex, which means that adult females of these mosquito species, which I'll talk about more in a second, can only be differentiated by ITS2. There's a molecular assay out there developed by Rick Wilkerson that can differentiate members of this complex. Despite the fact that they can't be differentiated, they have very different phenotypes in life history. So Anopheles bradleyi, like I said, is a coastal marsh mosquito. Anopheles crucians is more of an inland freshwater mosquito. Anopheles georgianus is a southeastern inland mosquito we don't have in New Jersey, but it is a member of the complex, along with several other lineages that people just call Anopheles crucians A, B, C, D, and E. Georgianus itself is added as lineage F, so that would make 7 ITS2 haplotypes of this Anopheles crucians complex, all with mosquitoes that can't be differentiated as adult females, but in fact are very different as far as their biology goes, and I think that's really neat. So it is possible to identify larval and pupal bradleyi using morphological characters, but it's not known at this time which of the other taxa might be all of those Anopheles crucians A, B, C, D, and E lineages. We don't know if we can differentiate those yet with any larval or pupal characters. They do appear to be reproductively isolated. When Rick and John Reinert agree on something, it's usually pretty important that there's no evidence of hybridization yet in this complex. So they could be separated by time, space, or otherwise. But this gave us a really cool opportunity to do a few things with our crucians mosquitoes. One is to assess genome evolution within the complex that could, for example, lead to halotolerance in a mosquito. If you think about crucians versus bradleyi. Another is a really fun question that we asked regarding response to changing environment and insecticide use. Right, so I saw Anne was on. Anne, this is the part where you can start paying attention because we're recapitulating this with some other things like stink bugs. So here's where the not necessarily pool seek, but where molecular evolution comes into play here. Using shotgun genome sequencing of these mosquitoes. Specifically, I'm going to talk about number 2 here. Response to changing environment and insecticide use. We all are also working on number 1. Number 2 was pretty fun. So I presented this at NJMCA last year, so apologize to the folks that already saw it, but it's quite interesting. So extant Anopheles bradleyi were sent to us by Mike Gustre from Atlantic County. He sent us an entire light trap worth of bradleyi. We only needed 1. We generated both Illumina short read sequencing libraries, we generated 2 sets of Illumina libraries. One was very high coverage data on the NovaSeq, 260 million reads. Another was some lower coverage, longer read data on the in-house MySeq, and also about 380,000 reads on an Oxford Nanopore single molecule sequencer that I have in the lab. This is a little USB sequencer. You just, make your sample, pipette it in the nanopore, and let it run, and it makes very long reads. So in this case, they were about 6,000 base pairs. So at this point, I was kind of just experimenting with different sequencing technologies. Luckily, there's an assembler that doesn't hate the fact that you use all these different sequencing technologies, which is Mazurka. It combines short read and long read molecular data in different ways. And the assembly wound up being really good. I was really happy with it, despite the fact that it was in 2,800 scaffolds, which, it didn't assemble the chromosome, and that's okay. We didn't need it to. It was 180.2 megabases. That's about the exact size of an anopheline genome that we want to see. The N50 was very big, which means half of our assembly was in scaffolds of 138 kb or larger. We predicted 12,900 gene structures on it, which again is right is on the bullseye for an anopheline mosquito, and found 94.1% of core arthropod genes were present. So that was our extant Anopheles bradleyi genome. Tahila was working in our insect museum, our Rutgers RUAC, Rutgers University Arthropod collection at the time, and really wanted to kind of work the museum into it somehow, and so did I, because as its curator, I want to see it published and get in the journal space out there. So I thought, you know what? We have some of these really, really old salt marsh mosquitoes that were collected 100 years ago before we started nuking the Jersey coast with insecticides and all sorts of other runoff and all the things that happen at the Jersey Shore. It might be kind of neat to try to sequence a really old mosquito genome and compare it to an extant mosquito genome and then see how they differ. Right. So this is our, Betty the Bradleyi or whatever you want to call her that we took from the RUAC. On the pin, it was identified as Anopheles crucians. But based on the collection information in New Gretna, I kind of figured it might be Anopheles bradleyi. We extracted DNA from it, and since this is over Zoom, you can't throw things at me. Yes, I destructively sampled it, but we had an entire insert tray of this mosquito collected at this site by this collector, so there were dozens of others to take its place. I don't like destructively sampling things, but I'm a mad scientist, so I did it. Sorry, and I'm the curator. We generated an Illumina library from this mosquito. And using the DNA extraction, we tried to barcode it to make sure that it was a bradleyi. We couldn't get a long barcode, I think it's 400 base pair, 500 base pair to work, but we did get a smaller 180 base pair piece of COX-1 to amplify because the DNA was old and degraded because it was so old. And we barcoded it as bradleyi. So we went ahead and made the shotgun genome sequencing library. We generated about 225 million reads. Average length was very short. Most of them were adapter sequence because that DNA had degraded over the last, well, it's not really 100 years, it was 1930. Sitting in the museum, the reads were very short, but that's okay because we didn't need to really assemble this. We just needed to map it back onto the reference assembly that we had generated from the extant Anopheles bradleyi. So we map all those short reads back onto the genome we have to look at the polymorphisms that are present in them. We identified 92,756 SNPs in gene exons. So these are fixed nucleotide polymorphisms between these two mosquitoes. We also used some tools to calculate FST values for the coding sequence or gene-wise or gene-specific FST values. That comes with a caveat, right? So an FST is a fixation index. So you could have one gene with one SNP that's completely fixed. At this site, it's all A's in this mosquito and it's all T's in another mosquito, and that'll drive the FST to 1 because it completely differentiates them. But it's based on a few number of SNPs. So that may or may not be generally helpful. And in our case, most of the FSTs that were 1 were driven by very few SNPs. So what we did was consolidate the number of SNPs per gene with FST values. And if we just jump down the list a little bit, there are some genes that kind of rise to the top here that had, a high FST and also lots of SNPs throughout the coding region of the gene. And we took a look at those first. So for example, gene 897 here, what is gene 897? So here's a short read mapping of those 75 base pair historic bradleyi reads onto the extant Anopheles bradleyi genome, and you can see all these colored lines here are the polymorphisms in the gene, lots of which are in the exons here. There were 13 polymorphisms in the exon, the FST was 0.92. What does this do? It's a sodium-coupled monocarboxylate transporter. So it just so happens that this particular gene is actually a really good biomarker of neonicotinoid exposure for terrestrial arthropods. 2 genes reliably indicative for neonicotinoid exposure, even under metabolic inhibition of cytochrome Ps by nicotinic acetylcholine receptor subunit alpha 1 and sodium coupled monocarboxylate transporter. So we thought, wow, this is really interesting. This is something, it's literally the first gene that rose to the top of the list, and it's already been implicated in some form of insecticide exposure. In this case, it's neonics. It's also linked to selective sweeps at voltage-gated sodium channels, which for most folks should need no introduction as far as insecticides go, which means that it non-randomly segregates with the sweep. So let's jump down to the second gene on the list here, G4102. What's that? That's a serine protease. Specifically, it's a K12H47 protease, which is a homolog of the Anopheles gambiae gene, AGAP003642, gambiae being the model or the first model mosquito. We have a very good idea of what all of its proteome actually does. FST was 0.9. There were 12 polymorphisms in the exons, and you can see them highlighted here. What does this do? So this popped positive on a microarray for pyrethroid-resistant Anopheles funestus mosquitoes from Africa. So it had a fold change of almost 2 in their microarray. This particular serine protease, this exact gene, AGAP003642. So after that, having examined those two genes in detail, what I did was take the top 5% of genes as ranked by SNPs per base pair of gene length. So I took the 95th percentile of genes that had the most number of SNPs in them, and I dropped them onto KEGG metabolic pathways, and I took a look at the pathways that had the most number of genes incorporated on them. So if there was some pathway that had lots of genes that worked in tandem to do something important, then that particular pathway may be plastic or open to molecular evolution in the face of time. One of the first things I noticed here was the TCA cycle, was a citrate cycle. So this is just cellular respiration ultimately leads to succinate production, which feeds oxidative phosphorylation pathway. So there were 4 enzymes present in the TCA cycle that lead to the oxphos pathway that were in that 95th percentile of genes ranked by SNPs per base pair. Oxidative phosphorylation itself is involved, for example, with sublethal pyrethroid exposure. We can see here glycolytic processes including carbon metabolism, oxidative phosphorylation, and the citrate, the TCA cycle, are indicative of an overall reduction in respiration post-insecticide exposure. So we know that both oxphos and the TCA cycle are involved in pyrethroid exposure and detoxification. So that study that I just referenced, here are those 4 genes that were part of the TCA cycle on their expression heat map. They're all there. The oxidative phosphorylation, there were 5 enzymes in the oxphos cycle here, located here on their heat map, that were all part of this in tandem with pyrethroid exposure. So to summarize that part, what did we learn from our 100-year-old mosquito genome? At least 4 independent lineages of the crucians complex were identified rapidly from sampling in New Jersey County. We compared genome data from a 95-year-old Bradley eye museum specimen and an extant Bradley eye. What rose to the top immediately was the sodium coupled monocarboxylate transporter and a serine protease as having a highly discriminatory index with lots of SNPs. Genes assigned to metabolic pathways that were involved in, for example, the TCA cycle and oxidative phosphorylation were included in that 95th percentile of genes ranked by SNPs per base pair, have accrued large number of SNPs between these 2 individuals. Now, the Jersey Shore is a very different place than it was 100 years ago, right? So can we necessarily say this is obviously in the face of insecticide exposure? And the answer is probably not, right? There are a lot of environmental toxins and runoff and fertilizer and things there that were not there 100 years ago. But I think it's pretty strong evidence that some molecular evolution is happening in the face of environmental change. Unfortunately, this was not a population level analysis. So sorry, I'm not going to grind up any more 100-year-old mosquitoes, at least of that species, and do any work as I would have if we were looking at, for example, population level. But I think this is kind of the best of what we can do with what we have. Until we can start generating entire genomes from a single 100-year-old mosquito leg, and maybe we'll get there, maybe we won't, I'm not completely sure. Okay, so where do we take this project in the future? We do have a shotgun genome sequencing library from a confirmed Anopheles crucians A that Tehila collected last summer. We did generate all of our Illumina genome data from it, and we can now compare an extant Anopheles crucians genome to an extant Anopheles bradleyi genome to begin assessment of genetic variation within the complex and start to get at some of those factors like halotolerance between crucians and bradleyi, for example. That data is in hand. It just needs to be analyzed. So to summarize everything that I've talked about thus far, I'm ready to wrap it up. I've spoken about what we do with regard to prophylactic measures to mitigate threats posed by vectors, particularly in this case, Hemiphysalis longicornis, to things like livestock by developing reverse vaccines using tick proteomes and tick genomes. Novel means of informative arboviral surveillance. So we're assessing things like pathogens as well as viruses that we didn't know were here or have been identified in North America or in the Americas for the very first time. Obligate and facultative associations with various microbes and ticks, things like Candidatus midichloriae mitochondriae question, and whether or not we may have an understudied pathogen in some proportion of our Amblyoma americanum ticks. And molecular evolution in the face of, for example, insecticide use within species complexes of mosquitoes, which I'd like to move forward on in the future as well, because I really like being able to use our museum for things like this. Okay, acknowledgement slides always stress me out because I never really know who to. There are more people than I could possibly put on this list, but the folks that were directly involved in things that I immediately talked about in this are here. Everyone from CVB, Dina Alvaro, Andrea, Emily, Heidi, Jim, Taiwan, and Environmental Sciences, Mehdi, Nicole, past and present grad students, Zoe and Steph, postdocs, Mohammed Mustafa, folks at USDA that have been incredibly helpful, Perot, Silao, Susan, Karen Poe, Kenan Oyen, Reggie Bastos, and some undergraduates that have done some really fun stuff in the lab over the years, Miranda Barnes, Tahila Jenkins, Fiona, and Eric. As is always, Nicole gets her own extra special slide for always being able to get stuff on and off of the sequencer and generating most of the data that we use to exist in science, and all of the funding sources so far, USDA, NIFA, the NJMCA, DEP, the NIH, Rutgers Office of Research, AMCA Research Fund, and the Hunterdon Health Foundation. I'm amazed because I did not practice this talk once over the last 2 weeks, and it's 11:58. So I hit the nail exactly on the head